Hello everyone, welcome to the PMRF-NPTEL session on the course Bioengineering. We will wait for 5 minutes till everyone else joins and start today's session. In the meanwhile, Niranjana, if you have any questions, then we can start with that in the meantime. Any doubts on the week 5 lecture course, in which we learned about chromosomal abnormalities and chromosomal basis of inheritance, as well as a bit on virus and bacteria.
No, sir.
Okay, let's wait for 5 minutes and then we will start. Let us get started and then we'll see as people join in. We'll let them in into the session. So, I welcome you all to the Bioengineering and Interface with Biology and Medicine NPTEL course, PMRF NPTEL session. This is the week 5 of the sessions that are being held weekly for this particular course. PMRF NPTEL sessions are interactive problem solving sessions in which a PMRF fellow such as myself interactively solves various course problems with students who have enrolled in the NPTEL courses. So this week we are going to deal with week 5, in which, as I already said, the discussed course material included chromosomal inheritance and chromosomal abnormalities, as well as a bit primer on bacteria and viruses. So let us get started. First, let me introduce myself. My name is Anubhav Chatterjee. I'm a PMRF Fellow and PhD scholar at the Developmental Neurobiology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. So, let us get ahead and look into our first question this evening. So, the question is, the column A represents an organelle's name, and column B represents functions of the organelle. Match the organelles with their correct functions. So the organelles given are nucleus, plasmid, fibri, sex, pilus, and cell wall. Functions given are uh, facilitates conjunction, basic genetic material, protection, structure, and selective nutrients to the cell, extrachromosomal DNA, and facilitates adherence to the substrate. The options are given here. These are round up of options, so only one of the options are correct. So I would like some answers in the chat. Like, you can guess whatever you think is the correct option. Niranjana says it is option number 3. Nucleus goes with basic genetic material. Plasmid goes with extrachromosomal DNA. Fibria with facilitates adherence in the substrate. Sex pilus is facilitates conjunction, and 5 cell wall. its protection structure, and selecting nutrients to the cell. Okay, so let us go ahead and go through the solution slide on this and answer the question for once and for all. So we'll go through various cell organelles and learn a bit about them before answering the question. So the first one that we are going to look into is cell membrane and lipid bilayer. So cell membrane and lipid bilayer is a very important functional component of the cell that divides the cell from the external environment. Now, looking closely, the structure of the cell membrane is the thin double-layered film of phospholipids covering the entire cell. So, what are phospholipids? Phospholipids are a huge fatty chain of huge lipid molecules which have a hydrophilic end and hydrophobic end. The hydrophobic ends are present in the interior of the cell membrane, and hydrophilic ends are present on the exterior of the cell membrane. So we have a double layer, that is one layer facing the exterior of the cell, the extracellular side with its hydrophilic heads, and the other layer with its hydrophilic ends facing towards the interior of the cell. And the hydrophobic ends are joined together in the middle of it to form the thick section, like middle portion of the cell membrane. So one end of the cell membrane is hydrophilic, that is soluble in water. The other end of each of the phospholipids is soluble in fat. Water soluble substances cannot penetrate. So this is obvious since the entire cell membrane is made up of lipid molecules which generally do not allow water to dissolve with it. So water soluble substances cannot penetrate through the cell membrane while fat soluble substances can. And there is also an important part to the cell membrane, that it is not only lipid bilayer but also has embedded glycoproteins. So proteins which have glycosylation post-translational modification in them, which is something that we discussed in the last lecture. And these can also be of two types. First is integral proteins that pass wholly through the lipid bilayer, and this can be ion channels, and peripheral proteins which are present on one end, either on the inside or the out intracellular end or the extracellular end. Present on the extracellular end, we have various ligand binding sites and cell receptors which can be present on one end, and on the other end we have their signaling partners such as GPCR molecules or G protein coupled receptors which bind to G proteins, and those act as the peripheral proteins. The entire membrane carbohydrate complex is known as the cell glycocalyx. So, as I said, that the integral as well as the peripheral proteins are glycosylated. So they have sugar moieties that are present and expressed on the surface of the cell. So this gives a very unique signature to different cells. So depending on what membrane carbohydrates are present, cells can be differentiated based on them. And this entire glycosylated complex of the sugar moieties present on the membrane of the cell is known as the cell glycocalyx. So many a times the immune system determines the foreign cells via their cell glycocalyx due to presence of foreign sugar moieties present on them, which are recognized by the antibodies as foreign and then destroyed subsequently. So this was the brief introduction to cell membrane and lipid bilayer. Next, we move on to nucleus. So most of the discussion here is being for cell organelles is being done on the eukaryotic cell because that has the most double lipid, which has the membrane bound complex organelles that we discuss mostly. So nucleus is one of those, and it is obviously known as the control center of the cell and contains DNA information in the form of genes, which are encapsulated in the chromosomes. During cell division, the DNA divides first into identical sets, after which the cell divides. The previous process is known as that the DNA division is known as karyokinesis, followed by cytokinesis, that is division of the cells. The nucleus itself is separated by the entire cytoplasm by the nuclear envelope or the nuclear membrane, which is another membranous body which allows very selective entry and exit from the nuclear pores. So this allows only selective proteins or other molecules to pass through into the nucleus, and this also is one of the steps in post-transcriptional regulation of genes. One other point is that in case of prokaryotic cells, we obviously do not have a nucleus. We have what is known as a nucleolus, and the chromosome just floats around in the cytoplasm. What difference it creates, the major difference that it creates is that in case of eukaryotes, the mRNA is being formed inside the nucleus first, the nuclear mRNA, which is not the mature mRNA. It is getting spliced and 5' capped, and then it exits the nuclear pore complex into the cytoplasm as the mature mRNA. And in the cytoplasm itself is the first time it is introduced to a ribosome, which starts the process of translation. So in case of eukaryotes, the process of transcription and translation have a gap between them. But whereas in case of prokaryotes, as I said, there is no question of any nuclear membrane. So the as and when transcription occurs, even if a small part of the mRNA is being made, a ribosome goes and sits on top of it. And this allows the translation of the gene into protein simultaneously to go on with transcription. That is, in case of prokaryotes, the process of transcription and translation are coupled. So there are very beautiful electron microscopy images in which you can see the entire chromosome of the bacteria, on which there are small strands of mRNAs being produced, on which ribosomes are sitting on top of them, which are forming the new proteins. So this was a brief introduction to nucleus itself. Let us move forward to endoplasmic reticulum. So the endoplasmic reticulum is another big cell organelle, and it is a network of tubular and flat structures, as we can see right here. It contains a huge cell surface area, which is 30 to 40 times the cell membrane area of the 30 to 40 times the area of the cell membrane itself, because of the folds. It is majorly involved in the synthesis of proteins and lipids. The proteins are synthesized mostly in the granular endoplasmic reticulum, which have ribosomes embedded into it, and the lipids are synthesized in the agranular or the smooth endoplasmic reticulum, which are on the other side. As I said, rough ER has ribosomes that are attached to it, and smooth ER has no ribosomes. So an important step in protein production occurs when once the protein is being produced, the ribosomes, along with the new protein that is being peptide that is being synthesized, goes and sits on top of the endoplasmic reticulum. After which the protein enters into the endoplasmic reticulum and undergoes various cleavages and post-translational modification as well as folding. After which they are released from the endoplasmic reticulum. So endoplasmic reticulum is very important for synthesis of protein as well as their correct post-translational modifications as well. Then we move on to the Golgi apparatus. So Golgi apparatus is famously known as the packaging center of the cell. So this organelle controls which molecule will be localized to which part of the cell. It has a very similar membrane complex, similar to the smooth ER. As you can see right here, they are very similar in nature, but it is farther away from the nucleus, and endoplasmic reticulum is very close to the nucleus. And Golgi apparatus processes cargoes to form lysosomes and secretory vesicles as well, on top of determining the localization of various cargoes. So the important factor in determining where a protein will go or be localized to is known as is the signal peptide. So if every protein has towards one terminal has a signal peptide that determines which location in the cell it will be localized to. So depending on what the function of the protein is, it can either be localized to the cytoplasm itself, or to the cell membrane, or might even have a role in transcription and be localized to the nucleus itself. So after being packaged in the Golgi, it will go back to the nucleus. Or if it is not well folded or not functional, then it will be directed towards the lysosomes, where it will be completely degraded, so that and the amino acids that are being produced from that degradation process be reused in the process of translation. So this is the function of the Golgi apparatus. So one of the important things to mention here is that even though most of the time we are drawing our eukaryotic cell or the prokaryotic cell like this, as if the organelles are floating on top of the cytoplasm, which is essentially a sea of cytoplasm, that is not the case. And these organelles do not have much movement intracellularly, as might be apparent from the diagrams that are here. Cytoplasm itself is very, very dense, which has been compared to the density of concrete in various papers. So the organelles do not have much movement, as in they cannot swim to their respective position or be localized. So if a particular protein wants to go to the cell membrane, it cannot just swim to the cell membrane. What actually happens is that there is an entire pathway or network of cytoskeleton, of the cytoskeleton, or that connects different parts of the cell, and our organelles sit on top of this cytoskeleton, and depending on the single peptides, a protein that has been packaged into a particular vesicle will essentially catch a train on this actin cytoskeleton backbone and reach it via the with the help of carrier proteins. So once it is synthesized here, it will attach to a actin cytoskeleton fiber and be transported via the carrier proteins, essentially taking a bus or a train towards its final location. So that was Golgi apparatus. And obviously we know about the mitochondria. Mitochondria is the powerhouse of the cell, and it is present in all areas of each cell, responsible for the major share of energy metabolism. But obviously it is a eukaryotic organelle again, not a prokaryotic one. It is one of the few two double layer lipid layered membranes, organelles having a double lipid layer membrane. So it has two layers, one inner, one outer. It also has a unique mitochondrial DNA and replicates autonomously without any help from the nucleus. And the matrix, or the internal part of the mitochondria, contains the enzyme that produce ATP. And further, it has been noted and is established that mitochondria, as well as the chloroplasts in case of plant cells, were initially external prokaryotic bacteria, which were engulfed by the eukaryotic cell and later passed on through the progenies, and then finally became the mitochondria. So mitochondria as well as chloroplasts have prokaryotic origins. And finally, we will discuss a bit on plasmids. So plasmids are extrachromosomal replicating double-stranded circular DNA molecules generally found in bacterial cell. So all of the points are important. First is it is extrachromosomal, that is, it is not part of the chromosome and can move around the cytoplasm. It can replicate autonomously, that is, it does not need the help of the nucleus or the ori site or the origin site present on the chromosomes to help it replicate. It can replicate on its own. It is double-stranded, that is, it has two strands. It is circular in nature, as you can see right here in this picture. And it is generally found in bacterial cells. Mostly mammalian or animal higher eukaryotic cells do not have our plasmids. They are extrachromosomal elements mainly present in bacteria. They are found mainly in the lower organisms such as bacteria, archaea, and protozoa. And they encode especially for antibiotic resistance genes, nitrogen fixation, metal resistance, and toxin production. So these plasmids have genes that help the bacteria to survive and produce extra proteins that are not essential for its survival. The gene product is not essential for function of the bacterial cell. That is, if you can remove the plasmid from the bacterial cell, it will survive even in specific conditions. But plasmids have genes present on them that give the bacteria essentially an edge to survive in nature. So plasmids are also extensively used in molecular biology and recombinant DNA technology, and they have been transformed into something known as vectors. Vectors are plasmids that have been manipulated and can be used to transfer essentially our gene of interest from one cell to the other. So one of the vectors are shown here, vector plasmids are shown here, and what so vector plasmids are of three types. It can be a cloning plasmids, which is used to clone our gene of interest and replicate it in large quantities. It can be an expression plasmid, which is used to express the gene of our interest, or it can be a shuttle plasmid, which is used to transfer a particular gene from prokaryote to eukaryotic system. Here on the particular slide, you can see a cloning plasmid. So, cloning plasmid is something that we will encounter mostly in our work. So, it contains majorly three, four things. First is the origin of replication, which is used for autonomous replication, an antibiotic resistance gene, which is used for primary screening of the bacteria, a selectable marker that is used for that is used as a reporter to mm, check for effective transformation of a bacterial cell. Promoter region, if it is contains a promoter region, then it should be expression plasmid instead. So and 3' and 5' primer binding sites, these are used for primer amplification of our inserted genes. Restriction sites are the multiple cloning sites that contains multiple restriction enzymes in which our gene of interest can be cloned into, and obviously our inserted gene that the vector carries. So essentially, this is the use of a vector plasmid. Then this will then be transformed into a bacterial cell, and then bacterial cell can then replicate and produce large amount of our plasmid. So this is how the entire process of cloning is done. It is based on these vectors. So transformation and cloning is something that we have already discussed in previous sessions. So we will not invest a lot of time on that. So that was our particular general introduction to how exactly different organelles in the eukaryotic cells work and what their function is. And coming back to our question on matching our left hand organelles to the functions on the right hand side, Niranjana was correct. It is option C, in which a nucleus contains the basic genetic material, plasmid is the extra chromosomal DNA, fibrae helps in adherence to substrate, sex pillars facilitates conjunction, and cell wall is for protection, structure, and selective nutrients delivery to the cell. So if anyone has any questions, we can discuss that before moving forward. Does anyone have any questions regarding the various organelles that we discussed, or anything else regarding this particular question? Okay, so we move forward to the second question, which is a pedigree analysis. So pedigrees, I think, is something that we are very familiar with. Pedigrees are particular schematic diagrams which are helped to show various diseases and their inheritance. So we are given a pedigree diagram right here and asked what exactly is our type of inheritance that we see. So we obviously know that the circle means a female, the box means a male, and if in this particular diagram a red circle means that the particular uh, entity is actually affected by the disease. So depending on the pedigree, please I would suggest everyone to try and figure out what the mode of inheritance here is. We have four options, all of them are circles, only one is correct. The options are Y-linked recessive, Y-linked dominant, X-linked recessive, and X-linked dominant. So I will encourage everyone to try and go through this. So what then it says, it is option D, That is, it is a X-linked dominant disease. So anyone else? Niranjana also says it is X-linked dominant. Anyone else wants to try and answer this? Shreya also says it is X-linked dominant, or option D. Okay, so let us go through the solution slide and try and come back to this. So there, here we have. Let us first go through the major two types of X-linked inheritances, and then try and answer the question again. So we have X-linked recessive first. So X-linked recessive diseases are ones which are carried on the X chromosome but are recessive in nature. That is, if there is a dominant copy of the wild type copy present, then they will be suppressed. But if a female which carries two X chromosomes has the X-linked recessive mutation, then they will be known as the carrier female, and they can pass on the particular disease to their progenies. So let us look at two specific cases what that can occur. So first case is that we have an unaffected father which has normal XY chromosomes, and a carrier mother which has a mutated X and a normal wild type X chromosome. The carrier mother is not affected by the disease but only carries the mutation. So if we cross them, we have if one of the cases can be that in case of the male, he will receive one X chromosome from the father, one X chromosome from the mother, and one Y chromosome from the father. So the male can we have a 50% probability of the male child actually being affected by the particular disease. So we have one unaffected son and one affected son, because he obtained a mutated X copy and no X, he does not have another X chromosome to but dominate over the diseased copy, and that is why he is affected. In the other case of in the case of females, she will have one X from the father and one X from the mother, and in this case also, if the particular female receives both wild type copies, they will be unaffected. If they receive one wild type and one mutated, then they will be carrier. So we have 50% chance of males being affected and 50% chance of daughters being carriers. In case of extreme recessive, if the father is actually affected, then we again have four cases. In case of males, there is no case of inheritance of the particular disease, because they will always receive the Y chromosome, so they are unaffected. But in case of a cross with an unaffected mother, we have 100% chance of the daughter being a carrier, because they will receive one copy of the mutated X and one copy of the wild type X. So this is in case of affected father cross an unaffected mother, then we have completely unaffected sons and 100% probability of the daughters being carriers of the particular disease. Any question from this particular inheritance slide? Okay. So we move forward to the X-linked dominant case. In X-linked dominant, we have a particular disease that is carried on the X chromosome, but it dominates over the wild type copy. So in case of the first cross that we saw, unaffected father cross and affected mother, then if we have again we have a 50% probability of sons actually acquiring the particular mutated X chromosome, and one of the affected sons will be directly affected by the disease, because they do not again have the X chromosome to another X chromosome to dominate over the wild type copy. So they will absolutely be affected. But in case of females, one daughter will receive the mutated copy and the wild type copy from the father, and they will be affected because one X chromosome mutation is enough for X-linked dominant mutations. But another daughter will be one which will have unaffected wild type X chromosome for the father as well as an wild type chromosome from the mother, and they will be unaffected. So 50% of the F2 progeny, F1 progeny will be affected by the particular disease. But in case of affected father, what we will have is the sons again do not receive any X chromosome for the father, so all the sons are unaffected. But in case of the daughters, we have an X chromosome, they always receive an X chromosome for the father, and they will be directly affected by the particular disease. So all the 100% of the daughters are affected and none of the males are affected. This is this discussion on X-linked dominant mutation. Anyone wants to ask anything regarding this? Okay, but then says no. So we'll move forward and try and answer this question. So we have our in the first case we have a female that is affected by the particular disease, and in the next progeny we have the females being affected by the disease as well. So this will be X-linked dominant. Everyone is correct. So this is marked wrong. So the answer given by everyone is correct. If the mother passes down a particular gene and all the females are affected, it will be X-linked. No, wait. No, it won't be X-linked dominant. I was correct in the first place. So see, what happens is that we have a chromosome coming from the mother, which is X-linked. So since we see X-linked chromosome being passed on, so one female is here which has a wild type copy from the father and a normal copy from the mother, and still they are affected. So we have one copy from the mother and they are affected, but one female is there which do not have any copies of from the which receive one wild type from the father and an normal from the mother, and they are not affected. Yes, so it is a extreme dominant because 50 we see in case of the mother passing down the gene, only the 50% of the females affected and all the males are affected. The answer is correct. So yes, in case of the father passing down, all the females are affected and the males are not affected. So the correct answer to this particular question is X-linked dominant. So we move forward to the next question. Uma marries Saket. Both of them have normal vision. Uma's mother is colorblind. What is the probability of the child to be colorblind? So the options are: all male progeny are colorblind, all female progeny are normal phenotype, half the female progeny will be colorblind, half the male progeny will be colorblind, and all progeny will be normal. Only one option is correct out of all of these, and this is circle type of answer, so only one of them is correct. So you can go ahead and try and answer this. So what the information suggests is that the parental generation should start from Uma's mother's generation. So one step ahead we have Uma's mother who is colorblind, and another hint towards that will be colorblindness is X-linked recessive disease, and when passed down to Uma, you see that Uma has normal vision, so she received a wild type X chromosome, and thus she is heterozygous for the color blindness, whereas Saket would be have a normal wild type X chromosome and Y chromosome. And now we have to see if how many of the children are colorblind. So the hint is color blindness is X-linked recessive. So I would like everyone to try and answer this. Okay. So Niranjana says it is A. All progeny, all male progeny are colorblind. All female progeny are normal. Vardhini and Shreya says they are C. Or half the male progeny will be colorblind. Okay, let us try and answer this. So as I said, the disease is X-linked recessive in nature. So we have, what the information we have is that Uma's mother was a carrier. So that essentially is the left hand case we see here. So if Uma's mother is a carrier, we have one daughter which is a carrier of the disease. So whom we have here. So this is this carrier daughter is essentially Uma. And then when we cross this carrier daughter again with a male, which is unaffected, which is the second generation. So in the next case we have this being Uma and this being the father. We have that half the male progeny are being affected, as the X-linked recessive we discussed. One son will have a wild type copy of X and Y chromosome, and he will be unaffected, and one will have a mutated X from Uma and a Y chromosome from Saket, and he will be the affected son. So the, as you can see right here in the diagram itself, 50% of the male child would be affected. None of the females will be affected, but one of them will be will be a carrier of the disease. So the correct option is C. Half of the male progeny will be colorblind or affected by the disease. Any questions regarding this? Okay. So we move forward to the next question, which is a match the following type of question. We have different chromosomal disorders on the left side and different genotypes which match to the chromosomal disorders. That is due to which particular genotype abnormality do we have this name of the chromosomal disorder. We have to match the following. So chromosomal disorders given are Down syndrome, Patau syndrome, Edwards syndrome, and Turner syndrome. And genotypes given are trisomy of the 13th, trisomy of the 21st, trisomy of the 18th chromosome, and monosomy of XO and the monosomy of X. That is only one X chromosome. The options are given here. We already have an answer. Vardhini says it is C. That is, Shea also says that it is C. Down syndrome is 21st chromosome. Patau is 13th chromosome. Edwards is 18th chromosome, and Turner's is the last one, that is monosomy of XO. Niranjana also says the same, that is, it is C. So let us move forward and try and answer this. So these are the common chromosomal abnormalities that are present. So this chart just answers the particular question. Down syndrome is trisomy of 21, that is 3 copies of the 31st chromosome. Patau syndrome is trisomy of 13, that is 3 copies of 13th chromosome. Edwards syndrome is trisomy of 18, that is 3 copies of 18. And Turner syndrome is monosomy of X, that is only one X chromosome. And Klinefelter syndrome is one of the other very important chromosomal abnormalities, is when we have 2 X chromosomes and 1 Y chromosome. So given the particular question, the correct option is C. Okay, so we move into the bacterial domain for a bit and go through gram staining again. Abhilash wants to learn gram staining. He wrote the protocol in random order. What we have to do is help Abhilash arrange the steps of the protocol documentation. So essentially arrange the steps of the gram order staining. The options given here are heat sterilization of the slide, safranin staining, grams iodine addition for affirmation of the staining to the cell wall, crystal violet staining, viewing under the microscope, and acetyl alcohol or ethyl alcohol decolorization. So so given here are the options. These are circular options, so you can just pick on the one that you think is the correct one. Vardhani says the option. Yeah, Shreya also and Vardhani, both of them say the correct option is A. That is the correct order is heat sterilization of the slide, followed by crystal violet staining, followed by grams iodine addition for affirmation of the staining to the cell wall, followed by acetyl alcohol colorization, saffron staining, and then viewing under the microscope. Any other answer we have? Okay. So let us go through quickly what gram staining is. So gram staining, so way earlier, when we did not have microscopy was not very well evolved, scientists were looking for various ways to differentiate between types of bacteria or classify them into different subtypes. So one of the person who looked at it was Graham, and he realized that depending on the cell wall characteristics of various bacteria, he can actually differentiate them bacteria based on a very simple staining procedure, and that is what he named as Graham staining. So that is what he has named as Graham staining. So what Graham staining essentially is when we use the properties of the bacterial cell wall in our different types of bacterial cell wall to stain the particular bacteria. So the first step we have is obtaining a bacterial smear on a slide or a plate on a slide, and here is where the heat sterilization of the slide would be. So essentially smear it and then heat sterilize the bacteria by putting the slide on top of the flame for a bit. That essentially fixes the bacteria onto the slide, followed by application of crystal violet stain. This is the primary stain. So this just stains the cell wall of a bacteria. So every one of the bacteria will essentially take up the crystal violet and be stuck. The crystal violet will be stuck onto the cell wall, and the entire slide will look as violet. Next, we wash the slide to essentially wipe off extra crystal violet, followed by iodine. So iodine has an important function that it is a mordant, which essentially allows the crystal violet to bind firmly to the cell walls of the bacteria. So essentially this is fixing the crystal violet into the cell walls. Followed by washing of the slide again to wash off excess iodine, followed by application of ethyl alcohol. So this is the important step and the major step in gram staining. So what happens is that gram negative bacteria, so one of them will be gram positive, one will be the gram negative. So gram negative bacteria, what they will do is that they will essentially they have two layers in the cell wall, one peptidoglycan layer, and another layer will be the normal cell wall layer. So ethyl alcohol essentially wipes off the peptidoglycan outer layer, and since our crystal violet was attached to the outermost layer, these particular bacteria will now have no stain on them. So the gram negative bacteria become unstained, and then we wash the slide to wash off excess ethyl alcohol and add a counter stain saffron, which then stains the uncolored gram negative bacteria with pink color. And when we finally wash the slide and view under the microscope, we will observe that the particular bacteria are now colored violet and particular ones are colored pink. And this was the basis on which the bacteria were first classified as gram negative or gram positive. Gram negative bacteria that have two different cell wall layers, whereas gram positive ones which have only one cell wall layer. We will look into what exactly the cell wall consists of in the later slides. So the correct order is heat sterilization of the bacterial smear, followed by addition of crystal violet, followed by iodine, followed by ethyl or acetyl alcohol, and finally saffron in counter stain, and then observing under the microscope. So this is the order of gram staining. Anyone has any questions regarding this particular slide? Okay. So we move forward. and answer the particular question. So everyone was correct in choosing A as the correct option. So the option correct order is heat sterilization of the slide, followed by crystal violet staining, followed by grams iodine addition for affirmation of the stain into the cell wall, followed by alcohol decolorization, safranin staining, and finally viewing under the microscope. In this particular question also, that the viewing under the microscope is only the last option in one of the options. So the last step in one of the options, so obviously you view under the microscope at the final step. So that was a quick shortcut to solve this question. So that was the question number 6. We move forward to the next one. Anyone has any questions regarding question number 6?
No.
Okay. So we move forward, and next is another question on gram staining. An antibiotic is applied to a gram positive and a gram negative bacteria. After incubation of 12 hours, it was observed that gram negative bacteria survived, whereas gram positive bacteria were killed. Which is the following mechanism of action of the given antibiotic? Which is the following possible mechanism for the action of given antibiotic? The options are: antibiotic acts on the media which is toxic to the bacteria. Antibiotic acts on the specific extrachromosomal DNA of bacteria. Antibiotic acts on the flagella of the bacteria. Antibiotic acts on the peptidoglycan layer of the bacteria. So only one option is correct with circular type of options. Niranjana, the option is says the option is number option 4. Antibiotic acts on the peptidoglycan layer of the bacteria. Vardhini and Shreya also say the same, that the antibiotic acts on the peptidoglycan layer of the bacteria. Okay, so let us move forward. And so essentially, the only difference between the two type of bacteria that is given in the question is that they are gram positive. One of them is gram positive, the other of them is gram negative. So let us first learn what exactly is the difference between gram positive and gram negative bacteria and try and understand what can be the possible action of the given antibiotic. So here we have summarized in a table what exactly is the difference between a gram positive and a gram negative bacteria. So as we had just discussed in the previous question, gram positive bacteria have only one or single layered cell wall, whereas gram negative bacteria have double layered cell wall. Gram positive bacteria have several layers of peptidoglycan. Gram negative bacteria have single layer of peptidoglycan. Gram positive have a thicker cell wall with 15 to 18 nanometers. Gram negative have 10 nanometers. They have a small periplasmic space. They have a large periplasmic space. No outer membrane present. They have an outer membrane made out of lipopolysaccharides. They return purple stain after gram staining. They appear pink after gram staining. So this is gram negative versus gram positive. Gram positive has the outer membrane, and gram negative does not have the gram positive does not have the outer membrane. Gram negative has the outer membrane made out of lipopolysaccharides. And here we have so the gram negative bacteria also have various porine channels that allow for the various proteins to be selectively passed on through the porins and move into the cytoplasmic membrane, which are then and the cytoplasmic membrane is more or less similar between both of them, just that gram positive do not have a second layer, gram negative have a second layer of cell wall, which are consist of the lipopolysaccharide. So an antibiotic that is applied to the gram positive and gram negative bacteria after incubation for 12 hours, only gram negative bacteria survive, whereas gram positive bacteria were killed. Which are the following possible mechanism of action of the gram negative of the given antibiotic? The obvious answer is peptidoglycan, because that is the only thing that is different between the two bacteria. Why peptidoglycan? Because in case of gram positive bacteria, disruption of the peptidoglycan layer disrupts the cell wall and allows rupture of the bacterial cells and lysis of them. Whereas in case of gram negative bacteria, the peptidoglycan layer is actually protected by the lipopolysaccharide layer. So antibiotic cannot get through the lipopolysaccharide to act on the peptidoglycan, and thus the gram negative bacteria are protected from the action of antibiotic, whereas gram positive bacteria are not. So The antibiotic acts on peptidoglycan layer of the bacteria. So we move on to some classic examples of genetic experiments. So here is one of the examples. Given here is a schematic of a type of DNA replication hypothesis. Which of the following statement is correct for the shown image? So we have one of the models of replication in which we have the original DNA being replicated as is on in the next generation, and synthesis of a light blue colored or cyan colored new DNA on the right one, and this continues in the next generation as well. That is, this original template of double stranded DNA is maintained, and we get a new double strand being synthesized, and similarly we have the same on the right hand side. So which of the following is the answer represents this particular model of replication? The options are semi-conservative replication, conservative replication. Uh, Evidence for this particular experimentation was provided by Mesensel and Stahl. Experimental supporting evidence was provided by Mendel. Only one of the option is correct. We already have three answers in the chat. The option that is given is conservative replication by all of them. So all of us say that it is conservative replication that is being shown right here. So let us try and understand the types of DNA replication to understand what is given here. So there are three. So in earlier days when it was not discovered which type of DNA replication actually occurs in nature, there were three hypotheses that were offered. The first was that of conservative hypothesis, in which we have a parental DNA copy which is maintained in the subsequent generations, and we get a whole new double-stranded DNA being made by the cell in every replication step. So in the second replication, we still have the original double-stranded DNA present here, and and all of the other strands are newly synthesized. The second was semi-conservative, in which one strand of the DNA is conserved in this next generation, whereas a new strand of DNA is synthesized. Complementary strands are always synthesized. Similarly, we have one strand that is of the original DNA and a synthesized strand of the new DNA. And this continues in the next one also. So we have one strand of the original and one strand of the synthesized, and similarly here, and two new strands being formed from the first steps alternate complementary strand. So this yellow strand in the first replication, which will be a new strand complementary to that, will be synthesized. So we have two yellows in the next one. And finally we have the dispersive model, in which we break which suggested that the entire strands are not inherited as is, but they are broken up and inherited in pieces as shown right here. So we have the parts of the original strand, as well as the newly synthesized strands, which are shown in yellow, and this continues in the next one. So these are the three particular models. So obviously, given the question, we know, given the diagram, that it is the conservative model that is shown right here. But can anyone tell me which is the type of replication that happens in nature? So which of the three particular replication that occurs? So Vardhani says correctly that it is semi-conservative replication. That is the type of replication that occurs in nature. Niranjana also agrees to the fact that it is semi-conservative nature. So the middle point, that is one strand is conserved in the subsequent replications, whereas the complementary strand is synthesized. Next question will be, which experiment was done in order to determine which of the three particular types of DNA replication actually occurs. So what will be the who were the scientists who performed the experiment to differentiate between the three particular strands, three particular types of DNA replication processes? So way back when we did not have any idea which particular type of DNA replication was the correct one. So there were various experiments being done, but one of them were the seminal experiment that actually defined what exactly the type of DNA replication is. So can anyone tell me what the who the scientists were who did the particular experiments? Vardhini and Niranjana both say it is the Michelson star experiment. In that particular experiment, they used radioactive nitrogen bases, the containing the N15 radioactive nitrogen base, which was given in the media, and depending on the incorporation rate of incorporation amount. Hello, am I audible? Hello. Okay. So the Michelson-Stahl experiment utilized radioactive nitrogen bases which had the N15 radioactive nitrogen in them as compared to the normally available N14 isotope of nitrogen, which is the normal one. What they did is that in the first parental DNA they allowed normal nitrogen to be there, and they shifted the bacteria to the after the like the first parental bacteria, they were shifted to the media containing the N15 nitrogen bases on N15 nitrogen. So in the first replication, the new strand synthesized only contained N15, and similarly in the second replication, in the second replication, the strands, again the new strands only contained N15 nitrogen bases. So what they followed this up with is that after the second round of replication, they went ahead and did something known as cesium chloride, ultracentrifugation. So ultracentrifugation is a technique in which we spin particular substrates at a very high speed, above of 70,000 rotations per minute or RPM for a long time, which allows for the separation at a very minute level. Even if the difference between the particular weights is very small, it is still we can still separate out based on the weight of the particular or the mass of the particular substrate. So after ultracentrifugation, after the second replication, they expected to see various bands. So they saw for dispersive, obviously we will see almost one band for dispersive after the ultracentrifugation. For the semi-conservative, we will have two bands, with one being smaller in subsequent generations and one being larger in subsequent generations. And in case of conservative, we will have very small first band and a very large second band which keeps on growing. So comparing the ultracentrifugated substrates at the first replication step and the second replication step, and looking at the bands, they were able to distinguish between the more different types of replication models, and they determined that it was semi-conservative model that was the correct one. So that was the seminal experiment that was done. But let us move forward a bit. So if this particular experiment you were to perform in present day condition, which or with all the equipment that we currently have, what procedures or what experiment would you design to find out which of the following types of DNA replication is possible? Can someone unmute and suggest an idea on how exactly we can separate the different types of DNA replication models? You can even write down into the chat if you want, like a modern experiment to separate out the different types of replication. So Niranjana says using radio-labeled base pairs and normal base pairs. So So that is kind of what they also use. They just theirs was a bit, I would guess, simpler, since you cannot radio label entire base pairs directly. So the problem occurs is Niranjana is their experiments was way simpler and elegant, because with radio labeling the problem is radio labeling is very hazardous firstly, and secondly when you are introducing a radio labeled substrate into the cell, it is mostly the directly the compounds such as nitrogen or basic media compounds such as phosphorus that are introduced as radio labeled, because the cell, even if you give it a radio labeled base pair, it won't incorporate into the nucleus directly. It will first break it down into subsequent moieties and then incorporate or form different things. So you cannot directly radio label a base pair. So I will give a hint regarding this. That one of the ways that you can think of is that what they didn't have in their time was fluorescence. So if we were to use fluorescence in this particular experiment, then how can that be done? So if we wanted to do the entire experiment with fluorescent uh, nitrogen bases, or uh, fluorescent nitrogen bases, or fluorescent nucleotides, then how would we design the experiment? Like what changes should be done to this? Because obviously the major advantage of fluorescence is that it is way easier to handle, not at all hazardous, and cheaper than radio labeled nucleotides. So anyone has any idea, if we wanted to use fluorescence to design the entire experiment, how would we do it? You can unmute yourself and like give your ideas. What can be the option? Like what different things that we can do, or you can put into it into chat. What do you think can be the use of fluorescence in this particular experiment? Okay, so use of fluorescence will be very similar to the use of radioactive nitrogen that they have done. You will just use in the in the first replication step, or in the first change of media, you will have nitrogen bases that are fluorescently labeled. So you have Psi3 bound to them or Psi5 bound to them, and let the cell use those. And in the first application you will have one entirely fluorescent chromosome, and in case of conservative, and another that is completely absent. In the semi conservative you will have both the chromosomes light up, and in the dispersive you will also have both the chromosome light up, but there will be a difference in the intensities. Similarly, you continue to the second step, you will have a huge difference as we go along in the third replication or so on, between the between the intensities between the three models, because obviously this particular one will have fluorescence throughout in all the particular cells or chromosomes, and this will not. And depending on that, you can differentiate or determine which exactly type of replication was the correct one. So now going back to a particular question, given depending on the schematic that is given, in which we have one double-stranded DNA which is carried over as is in next generation, and a newly synthesized strand is being prepared in the second generation, and in third one The original strand is again replicated as is, and we have newly synthesized two strands, and one of the original from the second one. And this is the conservative type of replication that we see here. So, any question that you want to ask regarding this? Okay. So we move forward to the next question, and this is a question again when we go back to genetics. In mice, the black coat color B is dominant over brown coat color small b, and a solid pattern capital S is dominant over the white spotted small s. Color and spotting are controlled by genes and assort independently. A homozygous black spotted mouse is crossed with a homozygous brown solid mouse. All the F1 mice are black and solid. All the F1 mice are black and solid. A test cross is carried out by meeting the F1 mice with brown spotted mice. That is, test crosses as we have seen between the F1 generation and the parental generation. So we have meeting between the F1 mice, which are black and solid, and the brown spotted parent mice that were present in the parental generation. And we have the first question with reference to this particular condition is, what is the possible genotype of the parents and the F1 mice? We have four options given here. You have to choose the correct one. What then it says it is B, that its parents are homozygous black and spotted, that is capital B, small b, capital S, and F1 is heterozygous of the four. Shreya also says it is B. Okay, so for this particular question, I will encourage everyone to sit down with a paper and pen, and try and jot down the Punnett squares on the pen and paper. So Niranjana also says, Shreya also said B. Niranjana is also saying B. Anyone else that wants to try and answer this? So, like, I would encourage everyone to try this particular question out, because genetics questions are very common in various exams. So, sit down with a pen and paper, jot down the Punnett square that are there, and this should be very easy to do for the first question at least. So, please try and do this. Okay, so we'll move forward to the solution slide and try and answer this question. We this what we have here is a dihybrid cross. So between black spotted and as given here, black phenotype is dominant, and we have a homozygous black spotted mouse. So black spotted should be capital B, and for the homozygous recessive we have small s. Similarly for brown solid, we have for brown homozygous recessive will be small b, and solid will be capital S. So this is our parental generation done. These are the genotype of the both parents. Next, we have cross between them. So as I have suggested before, in case of these dihybrid crosses, do not go ahead and draw the entire 9 is to 3 is to 3 is to 1 huge Punnett square. Instead, what you can do, which will simplify most of the questions, is get get hold of individual monohybrid crosses. So this is a cross between the black and the brown phenotype and the spotted and the solid phenotype. So separate them out and then cross them on a Punnett square, and that will be very easier to handle. So for this particular question, we have from the first parent we have capital B coming as the genes and the alleles, and the small b coming on the other side. You cross all of them, you have a heterozygous capital B small b. Similarly, for the second phenotype, second allele, that is spotted or solid, we have small s from first parent, capital S from the second parent. Cross all of them, we have heterozygous solid in the next generation, and this is the genotype of all the F1 generation animals, capital B, capital S, small s, and these are heterozygous black solid mice. So this was the entire Punnett square analysis. Anyone wants to ask anything from this? Okay, so this was the hybrid cross, and essentially we get at the end of it a heterozygous black solid. So this is the answer to the first question. What is the possible genotype of the parents and the F1 mice? So parents were, as we saw, capital B, small s, and small b, capital S. That is only possible in this particular answer. And also the F1 generation that we saw is heterozygous black solid, capital B, capital S, small s. That is given in the B option. That is all of you that have answered it in the particular chat are correct. It is the option B. So now we move a step forward with the question. Analyze the test cross results and select the correct genotypes and their expected ratios. So what was the test cross? Test cross is between the F1 mice, so capital B, capital S, small s, and brown spotted mice. So brown spotted mice were small b, small s. Uh, okay, so that was the brown spotted mice, and now you are being asked to find out what the solution to this test cross is. So this is the particular, what do you call it, test cross. So please, I would encourage everyone to try and answer this. This will be brown solid. Okay, so this has some problem in it. So the test cross is between brown spotted and black solid. So we'll do it together itself. The screen is visible, right? Hello. Okay. So for a test cross is done between a particular genotype and the homozygous recessive variant of the other one, the genotype. So we have black solid from the previous question. We have found out it is capital B, capital S, small s, and the recessive is brown spotted, which is small b, small s. So when we cross only the black and brown alleles, we have capital B versus small b. When we draw the Punnett square, we have capital B going with small b, capital B, small b on this first column, and similar in the second column. Capital B, small b, and small b, and small b. So these are the combination. So heterozygous and homozygous recessive that we get from the first cross. In the second cross, we have capital S, small s coming from the first parent, and small s coming from the second parent. So here is where the problem of autocorrect killed the particular question. So we have the crosses that happen are capital A capital A and small s. Similarly, we have small s here and capital S small s here. So we have heterozygous in both of them. So the entire combination, when we check amongst themselves, we have capital B small b with capital S small s. This is this one. Capital B small b with small s, which should be this one. Then capital B, no, small b with capital S small s, which should be this one. And small b with small s, it should be this one. So these should be the genotypes, and we see individually all of them have the ratio 1 is to 1 is to 1 is to 1. All of them are uh, genotypically have the same ratio. So essentially we did the entire problem alongside. So this is how exactly you solve the particular question. Figure out what the parental progeny is, look out for the particular allele, separate them out, and then draw Punnett square for individual of them. And then after drawing the Punnett square, just you have the genotypes in hand. So is this clear? Does anyone want to ask anything regarding this question? Like essentially we solved the question as is in the class itself. Anyone wants to ask anything from this? You understood everything, right? Yes, no, maybe. Hello, I am audible, right? Okay. So I will assume that everyone had understood what we were thinking here. So the correct option is option C. That is the correct option is option C. That is, these are the particular genotypes that we got, and the ratio is 1 is to 1 is to 1 is to 1. So I would like to ask a question on top of this. What do you think is the phenotypic ratio that we see here? So genotypic ratio is 1 is to 1 is to 1 is to 1. Ratio of them, all of them are the same. What is the phenotypic ratio that we see? Capital B. So this is essentially option E, that is the correct one. Niranjana says that the phenotypic ratio is also 1 is to 1 is to 1 is to 1. Anyone else wants to try? What do you think is the phenotypic ratio? Vardhini says also that the phenotypic ratio should also be 1 is to 1 is to 1 is to 1. Anyone else? One is to one. Okay. Shreya says the ratio should be 1 is to 1. Okay, so the phenotypic ratios are essentially you want to convert these to the phenotypes themselves. So black and spotted is what we will convert them to. First is due to dominant, it will be black and spotted. Second will be black and solid due to the capital S being the dominant one. Black spotted, black solid, brown spotted, and brown solid. So one of each four types. So the phenotypic ratio is also 1 is to 1 is to 1 is to 1. Okay, so that was that question. we come towards our final question, which is refer to the sequence below where the dot represents the centromere. So we have A, B, C, D, E on one side of the centromere and F, G, H, I. So this is a chromosomal sequence. So essentially these are genes is what you can assume them to be, and centromere is the dot in between. Match the chromosomal mutation with the correct sequence. The options are deletion, paracentric inversion, pericentric inversion, duplication and inversion. And the genotypes given are very varied, and you have to match which exactly the abnormality that we see here is compared to the sequence that we have on the right hand side. Only one option is correct. You can go ahead and try this particular question. Everyone is saying D. What Hindi? Do you have anything to say? Your mic is on.
No, sir.
Okay. Vardhini and Niranjana both say it is D. That is, this option is the deletion. A, B, no CD. This is a deletion. 2 part 1 is A, E, D, C, B, F, G, H, I. Inversion in one half of the chromosome. 3 with 3. No, I missed this. 3 with 4, where there is. Okay, so 3 with 4 is given here, in which we have essentially the entirety of one chromosome coming to the other, A and F, and everything else goes to the other side. And 4 is duplication and inversion, in which we duplicate our entire one part of the chromosome and invert it and add it. So that is goes with 2. Anyone else that wants to answer? Shreya says also that this is option D. Okay. Okay. So anyone else that wants to try and answer this? Okay. So the options given in the chat are the correct one. That is, it is option D. So let us discuss the various chromosomal mutations briefly. Chromosomal deletion occurs when we have deletion of the part of a sequence in the chromosome, so that just vanishes. So in this particular question, in this particular options, we have 3 as the deletion, in which the CD part just vanishes, and we have ABE in one arm of the chromosome, so that is the deletion. One goes with 3. Paracentric inversions are inversion that occur only in one half of the chromosome, like only one part of the chromosome above or below the centromere. So in this particular question, we will have just the inversion between one half. So that goes with part A, in which instead of A, B, C, D, E, we have A, E, D, C, B. Essentially, last 4 are inverted, whereas everything else on the second arm is safe. Pericentric is the one we have exchange between the two arms. So we have that we have in number 4, in which we essentially exchange the both arms. We have F that goes into the above arm and BCD that goes into the second arm, the lower arm, and that is number 4. And finally we have duplication and inversion, in which we duplicate, essentially mirror image our particular sequence and add it to the chromosome. That occurs in 2, ABCDE, and then we have duplication and inversion of EDC, and then followed by additional second arm being the normal one. So the correct option is 5D. Anyone wants to ask anything regarding this? Okay, I guess not. So that was the last question we had to discuss today. That is the so anyone. I would encourage anyone to ask any questions regarding today's session or any of the week 5 lectures. If you want to add anything, have any queries that we can discuss it now. Any queries you can unmute yourself or ask, or you can go ahead and put them into the chat box. Okay. So if we have no more questions today to discuss, then that will be the end of the session. So if no one has anything else to add, thank you everyone for joining today, and see you next week. If you have no more questions, then you can just leave the particular meeting. Thank you. Welcome.